The ancestral eukaryotes, which were known from fossils about 2 billion years ago, evolved a number of traits which all eukaryotes share today, such as a membrane-bound nucleus, internal membranes which will form the Golgi. They have endosymbionts, such as mitochondria. There are a number of genetic changes which they underwent, such as the cyclins and other proteins which control the cell cycle. They evolved a cytoskeleton using actin. And so I've given you a number of features that eukaryotes share. Eukaryotes owe at least some of their origin to endosymbiosis, where small prokaryotic cells evolved to live inside larger cells, and that these endosymbionts produced then mitochondria, and then in some lineages the chloroplasts of plants. We hold that mitochondria and chloroplasts are the descendants of endosymbionts because they are the size of bacteria, the shape of bacteria. They have circular DNA chromosomes like bacteria. They are like bacteria in their antibiotic sensitivity and how they make proteins and other factors. It was these early eukaryotic cells that first began to reproduce sexually. Now, although there are advantages to asexual reproduction, such as the rapid growth of a population, the ability to reproduce while by yourself, or not having to deal with mutations from a partner, the advantage of sexual reproduction is the great genetic diversity which it gives a population, and thus allows them to adapt to a changing environment. We had gone through details of the various groups of protists in class, and on the exam, some of the questions will be in the form of multiple choice answers, where you'll have the answer choices of the groups, such as diatoms, choanoflagellates, radiolarans and foraminiferans, etc. And then I'll simply ask those questions, which are in objective number 4. So if I ask, which are amoeboid groups encased in a hard shell, you would choose radiolarans and foraminiferans. If I ask which may have obtained their chloroplasts through secondary endosymbiosis, I'll ask for 2 examples, but there are more than 2. We think that dinoflagellates, diatoms, the euglenozoans, and brown algae obtained their endosymbionts this way, etc. You have a video of euglena, paramecia, and amoeba comparing how they move. Euglena using their flagella, paramecia using their cilia, and then the amoeboid motion as the amoeba reorganize their cytoskeleton and flow into it. I've given you videos of the reproduction of diatoms, where the outer frustule, which has those 2 halves, divides and divides, but in some of them it gets smaller and smaller to the point where now they have to form that auxospore, undergo meiosis, and then form a new frustule. And then I've given you a video on how paramecia reproduce, highlighting the macronucleus and micronucleus. And so I'll ask you to compare those 2, and so you should be able to give a summary of each of those. There's a video in which the features of the green, red, and brown algae are given. I'm simply going to ask you, to name 3 aspects that vary between these groups of algae. Finally, it is often difficult to divide protists into taxonomic groups because many have undergone endosymbiosis, and some of their genes of the endosymbionts have moved to the nucleus. And so when you're comparing a feature or a gene, one doesn't know whether this is the original organism or something which arose from the endosymbiont, which had a different history and origin, and so this can confound our ability to separate them into groups.